So Mendel's video notes for tonight, and the quiz will be tomorrow. So make sure you get yourself prepared. Take good notes. First of all, what do we need to know here? Okay, the person who is responsible for genetics and bringing it to us, introducing it to the sciences, we call him the father of genetics, is Gregory Mendel. Well, who is Gregory Mendel? Well, you probably heard some stories back in middle school. He was a monk, and monks back then basically lived a very isolated life where he was just at the monastery all the time, and led a very insulated, isolated life. So what he decided to do is he was a gardener, and he began to question how it was that the pea plant flowers were all white or all pink or all purple, and how that all came to be. So that's where he started basically his gardening interest, and how did flowers get that way, or how did plants get that way. So that's where he was at, and that's what he studied, the pea plants. All right, out of his work, and over a course of many, many years, we came up with three very prominent laws or principles, depending on what textbook you're looking at, that relate to genetics. The first one is the law of segregation. Well, you know what the word segregation means. It means separate, right? And that's exactly what happens. It's saying that the alleles that give us our different phenotypes have to separate during the process of meiosis, or you can't have the genes, the alleles that give you tall and short at the same time. You can't have the alleles that give you red hair and blonde hair at the same time. So that's what the segregation is talking about. The law of dominance says that we always have one phenotype or one allele that is going to be dominant over the other. So for instance, you probably suspect that brown hair is dominant over blonde hair, which is why there are more brown-haired people walking around in this world than blonde hair. It makes sense, right? That's the law of dominance. So there's always one that's more dominant than another. The law of independent assortment says that the alleles that give us our traits always assort or always separate independent of each other. Meaning, the alleles that give you your brown hair separate independent of the alleles that give you your brown or blue eyes. So your hair color doesn't necessarily give you your eye color, or your skin color doesn't necessarily give you your hair color. So those alleles that give us our traits assort separately. So you could be red hair, blue-eyed, which a lot of people are, and we're going to talk about that later on, or you could be red-haired, brown-eyed, or you could be blonde-haired, brown-eyed, or you could be brown hair, blue-eyed. There's no specific trick there. They assort independent of each other. All right, next slide. Where's my little doohickey thingy? There it is. Couple important things to take out of here. First of all, the labeling of the generations. P stands for parent, and it's referring, to that first generation that you're looking at. F1 is your first generation after the parents. F2 is your second generation after the parents, and then of course F3, 4, so on, so on, so on. Okay. We also need to look at what true breeding is, and true breeding is when plants self-pollinate each other as opposed to being pollinated by other plants. What do you need to know about? This is where Mendel started. So Mendel started by having a purple pea plant and a white pea plant, and when he crossed them in a pollination, and bear in mind, purple plants are purebreds and white plants are purebred. Okay, that means they're kind of all whole, right? He got all purple flowers in his first generation that was produced. This puzzled him, as you can imagine, because there were white flowers to begin with. Why did he get no white flowers? And that's kind of where the whole inquisitive thing came about. So he decided to cross those two purple flowers in the next generation, and look what happened. White reappeared. So, of course, now he's really got to figure this out because he's very confused, and so he keeps going with his experiments, and over a long period of time, and if you look at his data in the next slide coming up, you'll see that he definitely was at it for a while. But purebreds are, species that are not mixed. And then, of course, we know dogs are probably the most obvious situation, dogs and cats. You have purebred labs and purebred schnauzers and stuff like that. If you mix a lab and a poodle, you get a Labradoodle, right? Which is kind of a designer breed right now, but that's not a purebred. That Labradoodle is a hybrid dog mixed between the Lab and the Poodle. But now, if we want to keep that designer breed, if we take the Labradoodle and we made it with another Labradoodle, that's a purebred situation, and we're making Labradoodles. So hybrids can become purebreds at some point too, if that makes any sense. All right, this is his data. So you can see he got lots of pea plants in his time that he was gathering. And he looked at all different kinds of traits when he was doing his experiments. He looked at the shape, the color, the size, the position on the plant, how high the plant grew, and everything. Okay. Memorizing his data obviously is not important, but noticing that he put a lot of time into it and he got a lot of information out of it, and this information then sat idle for a long period of time until other scientists later, centuries later, decided to continue to investigate it, and that's when our laws of segregation, a law of independent assortment, and law of dominance came to be. All right, what else do you need to know about the chromosomes themselves and their features? Well, think back to transcription and translation. We took a segment of the DNA and coded it into messenger RNA, and then we made it into a protein in protein synthesis. Well, what was that code that we were talking about? Well, it was a gene. Okay, it's a gene segment. And what did it ultimately lead to? The production of a protein. So that's what we're talking about. That's how those two are linked together. Within the chromosome, there's a couple pieces that you want to know. We already talked about homologous chromosomes. You get one from your mom and one from your dad. And that's true from any sexually reproducing organism, okay, plants included. One of the things is that where the position of where those genes are located are called a locus, or a point, right? That makes sense. Kind of had a location, comes from that locus point. And then alleles are what give us the physical traits that we see, our brown hair, our blue eyes, our white skin, our black skin, whatever it is. Okay? Those are some vocabulary words that you've got coming, alleles. All right. Homologous chromosomes and homologous alleles, they're identical to each other, which we've seen that, right? So we already know the word, the prefix homo versus the prefix hetero, same versus different. So just apply what you know with your prefixes and you get the words here. Heterozygous means different alleles. So a homozygous alleles, meaning you're going to have the same allele. In this case, the allele is coding for wrinkled pea pods, and so you can see you got wrinkled from the one parent and wrinkled from the other parent. It gives you the pair of alleles. And if you remember back to middle school, wrinkled, we use letters to indicate this, and a lowercase letter means that it's recessive, and an uppercase letter means that it's dominant. So hopefully that's ringing a bell from middle school when you did Punnett squares. Heterozygous means different alleles, right? So in this case, we have one allele that came from one parent that was wrinkled, and one allele that came from the other parent that was round, and round is the dominant in this case. All right, so that is talking about alleles. And this is just explaining that dominant versus recessive allele thing again, and how you get two copies from your parents, how it's represented by uppercase and lowercase. Hopefully this is all bringing back memory. Remember the words genotype and phenotype from middle school? Genotype is what gives us the genes. Okay, so when we talk about our genotype, we're talking about the genes that actually give us the trait. The phenotype is how those genes are physically expressed. So they kind of make sense with the words, right? You see the word gene in there, and you see the word maybe physical starting there. Okay. Next slide. Punnett squares. Remember the Punnett squares. Okay, when you were in middle school, you did monohybrids. We are going to look at monohybrids, dihybrids, and trihybrids. So what do? You probably starting to remember this. Each parent is going to have a pair of alleles. Okay. In this case, the parent has a dominant and a recessive, and then the other parent is a dominant and recessive. And you mix them by bringing them down and bringing them over to make the different genotypes, and then the genotypes lead to phenotypes. If you have two capital letters, that means it's dominant homozygous, right? If you have one capital, one lower, it means it is heterozygous. But because of the law of dominance, the dominant trait, whatever it is always going to be expressed. Here again, same scenario. Two lowercase alleles is going to give you the recessive characteristic. So just for instance, if this was coding for blonde hair versus brown hair, this would be a brown-haired person. Heterozygous would be brown because brown is dominant, and this would be your blonde hair. Okay. And we do a test cross, which is a vocabulary word. We do a test cross when we don't know one of the parents' original genotypes. What we do is we mate that parent with a known recessive, because that's always expressed in the recessive form. In this case, it would be mating somebody who was blonde with someone who is brown, and seeing, are they brown homozygous or are they brown heterozygous? And I'm going to let that one kind of play out as a bell ringer this week, or shortly after spring break. So we'll see how that plays out in that kind of a test cross. All right, we're coming to the end, guys. Monohybrids, here's a couple examples, and we're going to go through these in some details as we prepare to do our worksheets. So I'm just going to let you kind of look at them. You can see you had a homozygous recessive white flower mating with a homozygous purple flower, which is dominant, so that's why it's in capital letter, and this is recessive, so that's why it's in lowercase. What did you get for phenotypes? Well, you got all purple flowers, and they're all heterozygous. If you take heterozygous and heterozygous, okay, so two purple flowers, but they carry the recessive allele, you get 75%, or 3/4 of your flowers are going to be purple, and only 1/4 that will be recessive for that trait. And these are tried and true. So these are tried and true, and you can do this, and you will see that the outcomes are based on probability. It will happen. It's a pattern for sure. So you take a heterozygous and a homozygous recessive, and you mate them, and you get a 50-50 split. Okay, so you can see how that plays out. Got a few worksheet activities that we're going to do, so we will talk about those more tomorrow, or you'll get into it on Friday. Last thing, I think this is the last slide. Yep, that's the last slide. So just want to point out to you what a dihybrid cross is, because in monohybrids you're only comparing one trait. In a dihybrid cross, you're comparing two traits. So you can see here, we've got a Y allele and we've got an R allele. And the Y allele is coding for the color of the seed, and the R allele is coding for the shape of the seed. So yellow and green are the two colors. Obviously, from just looking at this, you would suspect that yellow is probably dominant, right? And green is recessive, because just by the numbers. And then R codes for round or wrinkled. Well, which one do you think is dominant? Well, if you look at it, there's more round pea pods than there are wrinkled pea pods. So round is dominant, and wrinkled is recessive. And this kind of really demonstrates the law of independent assortment, because it says the seed color basically does not rely on the shape of the seed when aren't tied together when you when you do a cross of the alleles. But there is an exception, and it's called linked genes, which we will talk about in our advanced Mendelian genetics coming up after spring break. So there's your notes. Oh, wait, I forgot one more important thing. In a dihybrid cross, and this is something you just want to put to memory, if you have a heterozygous parent for both alleles and a heterozygous parent for both alleles, so I've got both heterozygous parents, right? I will always get a 9:3:3:1 phenotypic ratio. So if you were to count these up, you will always find that 9 will be dominant for one allele, and then recessive for the other. So it plays out in a 9:3:3:1. And we're going to do a dihybrid cross, so you'll actually see this play out in class probably on Friday. All right, now I'm really done, and I will see you in class tomorrow. Have a great evening, everybody. Take good notes.